morning. I'm Nicole White from the Trace and Environmental DNA Lab at Curtin University. I have Dr. Matthias Saccò and Dr. Josephine Hyde, and we've kind of got the little environmental DNA section of today's wetland and today's talk. So hopefully by the end of this session you're going to get a good understanding of what is environmental DNA, how do we do it, and how has it been applied. So case studies for conservation management and other applications as well. So I'm going to get underway. So what my task was today is I kind of get to give you the overview of what environmental DNA is. I don't know if you've heard the term environmental DNA before. Yes? Great. That's just a little bit of what the talk's going to be about, but I've kind of just introduced that to you already, so I don't think I need to spend much more time there. So we have this eDNA, and it's kind of been a bit of a buzzy term. So what is eDNA and how do we interpret what's environmental DNA? So environmental DNA is all the DNA that we can harvest from what we consider an environmental sample or a mixed substrate. So as you know, we all shed DNA, all organisms shed DNA constantly into their environment. So we're all leaving DNA behind in this room. Yeah. So technically we could do an air sample in here and then pick up all the people in the room because you're shedding your DNA. So that's what it was. That's what environmental DNA is. So it doesn't really matter what the source of the DNA came from. That's what it is. Now, before the term environmental DNA kind of came in, we were using the term metabarcoding. Right? So that means we take a mixed biological sample, I'll get into it more later, but that's where you can identify multiple species from one sample. And that's metabarcoding with a bulk sample. As you can see this little infographic, and this is kind of the types of projects and areas that our lab has been involved with over the years. We've been doing metabarcoding now for almost 12 years. And we've been fortunate enough to get into a variety of projects. So we're below ground, we're above ground, we're terrestrial, we're marine. We do different applications depending on what it is that we're asking of the DNA or what the study is about. So that's what I mean by what's the study about. So are we only interested in saying, all right, so we need to detect an invasive marine species in this area, and that's the only thing that we're actually interested in finding from this seawater. So there we would go about then actually designing an assay, something that is specific for that invasive species. This is what we call looking for the needle, because remember that invasive species DNA is going to be in that mixed sample. There's also going to be fish and crustaceans and marine microorganisms, all of that in that mixed sample. But in that mixed sample, we're saying, is the invasive species DNA present in it? So this is where we call the needle or the haystack. The haystack then refers to, well, what is in this marine environment or what is in this wetland? So it's more a biodiversity survey. So we can use environmental DNA in different applications in what we're interested in, and it really just depends on what genetic markers or molecular magnets, if that's an easier term, to understand, to pull out if the DNA is present in the sample. Now, the big kind of leaps in technology has allowed us to do this, and it's really actually made some leaps and bounds in the last 10 years. So before this next generation sequencing, high throughput sequencing, there's kind of some fuzzy terms going on. Before we could get a DNA sequence, which is something that we see at the bottom here, that's what we do. So we submit a sample off, we read the DNA sequence. That would be for what we call a single source sample. So example is you get a piece of fish tissue, you don't know what that fish is, you extract DNA, it's only that fish, and you generate one sequence. This is older technology, which was called Sanger sequencing. If we applied that technology to a mixed sample, we would end up with something like this, and this is the beauty of this next generation sequencing, is it allows us to sequence hundreds of thousands of DNA strands at once. So we can get the composition going there, and this is really the leaps and bounds in the technology. So how do we know what belongs to what? This is just basically a simple workflow of what it is that we do in the lab. To make it easy, every DNA extract at the above, so say this was seawater, this was dirt, this was an air sample, whatever, this was a scat, you want to see what predators are preying on in their environment. We extract DNA, it's a mixture. What we do is we kind of put on these magic license plates, so to speak, that we stick on the ends of the DNA that are coded to that sample. We run it through the high throughput sequencer, which can generate us anywhere from 1 million to 100 million sequences. But then what do we do with those 100 million sequences, and how do we know which one came from which sample? Is we can, from the license plates that we've added on the ends of the DNA, we've assigned those to each sample, so bioinformatically we can bin the sequences to the sample. Once we bin it, we rely a lot on supercomputing, because then we just have a DNA sequence. How do we know what the DNA sequence is? We've got to compare it to a database of known DNA sequences. So we rely a lot on blasting every single one of our sequences to the database, and hopefully we get a hit or a match, and we go, yes, we found that invasive species in sample A. That's a basic overview, and that's what we call the metabarcoding workflow. Now, something to be really conscious of and careful of, and we are a lot, we are very aware of this in our lab because we've been doing this for quite some time, is sources of contamination. And I really can't stress this enough, because sources of contamination can come in at any point. So contamination sources start right in the field during sample collection. And that comes with sharing of equipment just during different areas if you haven't properly decontaminated. Then you can falsely interpret your results that you found invasive species in X number of sites, but really the invasive species may have only been at site 1, but you didn't clean your equipment, so therefore you carried the DNA through across all the samples. Extremely, extremely important. So we have a lot of controls that we put in place when we're doing environmental DNA work, and that starts right in the field. Very, very important. The reason why I highlight this is basically what we're seeing now, and some of this article can't see it, but this was published in 2017. So microbiome research, that's on microbes, soil work. Up until then, there wasn't a whole lot of controls that were put in place, and what we found out now is a lot of people were publishing contamination results. So it's really stringent now with doing environmental DNA work and high throughput sequencing work. So it is, and Mick Watson, who's in the microbiome, said you need to know where your sources of bias and error are in your experiment. Otherwise, you're going to be interpreting results which you think may be your results, but it may also be contamination, whether that's come from the field or in the lab. So we're very fortunate at Curtin University. We have some awesome facilities, and I'm on a national working group that we're writing best practice and standard guidelines now for environmental DNA. And the basic minimum is this kind of clear separation of your pre and post PCRs. We get to go one step above that, and we have an ultra clean environment. So this is me when I go in and work in this environment. Because we also do ancient DNA work, so our background with environmental DNA comes strongly from ancient DNA, because we work with material that's very old, doesn't have a lot of DNA in it, and is highly subject to contamination. So this suited, the environmental DNA suited us very well because we already had workflows set up for ancient DNA. It kind of just plugged in. And this environment here is all HEPA filtered, and we keep everything clean. And then we have our modern labs, which are clearly separated from here, and we dedicate certain activities, and we have a sort of workflow for the day. So we don't go into our modern labs and then go into our clean environment. So we would be back. So what we do and the care that we take has resulted in quite a few years of developing this and what we do in the lab, but we stand by our data sets and that they're very high fidelity. So that's just a little quick overview on environmental DNA. I'm going to pass it over to Mattia, who's going to share some more exciting things, which is actually some results of some studies and some science.
Yes, I missed what PCR stands for.
Polymerase chain reaction. So that's the photocopier that we put the DNA in with the markers, and it amplifies it up. Yeah.
Okay, we have 5 minutes left, so I'll try to be quick, but not super too quick, for you to understand what we're talking about. So one of the biggest projects we have on Trend is the so-called EDGES. EDGES stands for eDNA for Global Environment Studies. The main goal is to develop and apply eDNA techniques to address biodiversity loss and sustainability. This applies worldwide because we have 5 different projects targeting 4 different environments: terrestrial, subterranean, marine, and freshwater. Today I'll be talking a bit about the freshwater one, given the topic of the conference. The title is Functional Ecology of Chilean Precordillera Lakes towards the Wetland and Index for Birds. So we're talking about Chilean Precordillera. Just to give you a brief overview, hypersaline systems refer to wetlands where the salinity is above 35 grams per liter. There are certainly very charismatic ecosystems around the world. You might know Salar de Atacama, you might know Braccio Lake, Lake Eyre in Australia. What's really interesting is that once we have a look at the total volume of saline and hypersaline lakes, they account almost for half of the inland water we have on our planet. Despite, despite the progresses that we've had over the last 10 years in hypersaline ecology, there's still a lot of question marks which are unsolved. The graph on the left is showing you a little bit of ecological flows and energy dynamics in hypersaline systems, but we still don't know much about the mechanisms sustaining biota, integrating water and also geology. Some of the main points that still remain unresolved are energy flows and nutrient loads, the migration of birds, and evolutionary patterns characterizing the microbial community. So, hypersaline systems also provide very valuable ecosystem services. Lithium, we all have lithium in our pocket. Artemia, which is driving the aquaculture industry, ecotourism, and recreational activities. These systems are hugely under pressure, also because there's a massive lack of social awareness of the importance of the systems. These two articles, recent articles, are talking about very key topics like climate change combined with the resources we have of these systems. And there's, if you click, there's the latest article, I think it's a month ago, about the big lake down south here in Western Australia, if you want to have a read about that. So 4 main aims in this project. First, we want to survey water and sediment both in space and time for prokaryotic and also eukaryotic taxa. We want to conduct hydrological and biochemical investigations of the surface water table. We'll be focusing on the Artemia, which is a brine stream that is worldwide spread and really important in terms of the ecological function of the systems. And last but not least, we will focus also on birds, qualified birds characterizing the systems. Now, we'll be targeting 3 lakes. You might know one of them, Atacama, and we pick them in the area of the Flamenco's Natural Reserve, which is Ramsar site. The nice thing about this sampling design is that it provides us with an altitudinal gradient, so we'll be able to detect spatial patterns. But not only that, we'll also be focusing on 2 consecutive seasons, so we will combine spatial analysis with also temporal patterns. The overall goal is to provide conservation analysis with the so-called wetland health index, which is considered more as a protocol by combining different disciplines rather than a raw number on the ecological value of the systems. I won't go much in detail about the analytical design, but as you all know, COVID had a huge impact as per every international project. So at the moment, if you click, that's the beautiful button. At the moment, the project is on hold. But this forced us to think out of the box. And if you click and go to the following slide, this forced us to think, hey, what can we do meanwhile? We want to test the use of this tool, and we want to actually have an idea on the applicability of this tool. So we went to Rockmist. Why is that? Because there are hypersaline lakes, similar hypersalinity to the Chilean desert. There's Artemia, there's wading birds coming all the way from Siberia, and overall the ecological functioning is actually quite similar. So we went there, we collected samples from 5 lakes. You might know, Vincent, Baghdad, Herschel, Garden, and Serpentine, and we collected a few water samples with the idea of targeting the microbial community. We also collected probe data, classic pH, DO, and other type of data, and we also did some nutrient analysis on the load of the systems. 3 main goals: test the eDNA, investigate the microbial community, and also assess the energy flows and metabolic patterns in the lakes. Very briefly, hydrochemical background. From the beginning, we realized that we were dealing with 3 main clusters: Garden Lake, the least freshwater and saline, but also the one with the poorest nutrient load. Then Baghdad and Vincent were clustering together, and the most saline systems were Herschel and Serpentine. We also did some fluorescence analysis in order to understand the nature of the carbon compounds in water, and we found out that Vincent and Baghdad were the only one which developed aromatic protein-like and human fulvic acid-like organic material. This is probably in line with the high volume of earth, the high volume of earth that you can find on South, having a role in increasing the carbon load in the system. In terms of microbial diversity, as I said before, we collected water samples from few sampling sites. To give you an idea, we're talking about 5 liters per each sampling site. And then we did some magic, some eDNA magic, and we targeted a widely used primer for studying the microbial community, which is the bacteria 16S. Through some bioinformatics, we got these graphs, which are actually interesting because the pattern that we observed through the eDNA targeting microbial communities was actually matching the clustering that we saw in the previous slide. There's 3 clusters: Garden by itself, and then we have Baghdad and Vincent, and then the other 2. What's interesting is that overall, the microbial community of these 5 lakes span from moderately to extremely halophilic groups. And TDS, which is the indirect way, probably also the most accurate way to test salinity, and also alkalinity were the main environmental drivers shaping this clustering. We went a bit beyond, and we tried to understand the functional patterns through this platform. This is not the best platform in the world, but it gave us a nice insight that can be explored further with more specific techniques. And again, 3 clusterings confirmed. But what's interesting from a functional perspective is that there's, if you click, there's increasing trend, which is matching the increasing salinity with more methanogenic and more aromatic degradation pathways. This is a graph pretty much summarizing what I'm talking about. The increasing salinity, there's an increase in methanogenetic patterns and different microbial diversity, while Curtin stands over there, which is the poorest nutrient, just to remind you. And from a functional perspective, there's sulfate and nitrate reduction. And more specific aspects that we are not, unfortunately, don't have time to focus on. I'll finish this part and then Josie will talk a bit more about the upper levels in the biotic community, so she won't talk about microorganisms. The idea is not to stop here. We want to expand this and we want to target actinians. Actinians are actually feeding on microbes and they're crucial for the systems. Actinians are also crucial because birds are coming all the way from Sevilla, also because actinia are providing a nice food source. So overall, the broad idea is to use eDNA, combine it with biochemical approaches to try to understand what's the functioning of the hypercellulase around us. To conclude with a positive note, our partners, collaborators in Antofagasta were able to collect samples. We had support from the Mars-like environment, which of course very challenging for them, very low temperatures. I've been talking about, as you saw before, above 4,000 meters above the sea level. And also we have a few ideas still in progress, but we are confident that it will happen about citizen science projects in order to raise awareness about the importance of the systems and also to create valuable data set for the scientific community. I'll finish here and Josie will talk a bit more about this project.
Okay, so I'm going to be talking you today about how DBCA is using eDNA. And most of the work I've presented today is most of the work has been by a master student I had with me last year at a student institute that's here in here. So I don't think I need to tell you all that freshwater biodiversity is declining, and that 84% of global freshwater species populations were lost between 1970 and 2016, which is, and most of this has been driven by a decline in habitat degradation, pollution, overexploitation, climate change, and invasive species. And this is all on here in WA. So to help conserve freshwater habitats, we need to measure a number of things, and from my perspective at DBCA, the things that I'm concerned about is what is actually present in these environments, and to discover invasive species before they become established or we can actually do something about them. So the project was focused on the Canning River. The Canning River provides an essential service to us here in Perth in terms of water, for industry, recreation, and is home to a diverse group of species. So there has been widespread degradation along the Canning River. This is due to a number of factors: pollution, climate change. There's been a lot of dams and weirs that have been introduced, and there's a lot of invasive species as well. So we need to protect and restore the Canning River from DBCA's perspective. We need a lot of information to inform its management, and this management also comes under this one. Canning River Management Act, if anyone was interested. So the method for this project is that we picked 10 sites between barriers, and we did 8 eDNA samples at each site. We also did fyke netting, but I won't tell you about that today. And where we picked our sites, where all the blue dots are our sites, and the red triangles were where the barriers are. And we did 9 for the fyke netting, and then we did an extra site here up in Rich, because that was where everything to the left of that, we found cichlids, which we were really interested in as an invasive, and everything to this side, we found very cichlids. We thought maybe we can use eDNA to sort of map the invasion threat, really optimistically. Okay, so next slide. We found around 58 species for a given definition of species, identified from 5 different classes. We were using fish primers, so we were targeting specifically fish, but sort of nicely. We also found a couple different other groups. But overwhelmingly what we found were fish sequences. The next biggest group was birds, and then mammals, gastropods, and reptiles also found. So next one. So yeah, as you can see, this is my fish group, regular fish, and that is the biggest group. Each graph has a different scale, and it has the most diversity. The next one was birds, and that was mostly ducks, and ibises were the two big groups. Then mammals, that was kangaroos, possums, racali, yay! Then dogs and humans. Yeah, and then for reptiles, the big pink one was skinks, there's a tiny little green one, and that was geckos. And then the gastropod, which is really interesting, is just one species. We got it down to species level, and it's an invasive freshwater species, and we only found it at one site, big low numbers. We're really hoping that that means that we might have caught it early enough to actually do something about it, but that's deep and not us, so yeah. Next slide. So if we just look at some of the fish that we're actually really interested in, so we have our pearl cichlids, so unfortunately we can't really map the invasion for it because it looks like they are doing a whole lot. Then goldfish, carp, and what we thought was just the western mosquitofish turns out to be also the eastern mosquitofish, and the eastern is more dominant and more aggressive, so that's that. But the western pygmy perch is also, really dominant, which is kind of yay for them. I'm not really that's when I come from the mosquito microbiome. So I'm really learning about vertebrates, and it's been great. So yeah, but the other thing we learned, if we go to the next slide, is that what we thought was the barriers might not be doing anything. If we actually look, it seems like maybe some of the barriers might actually be playing a role, and that there are multiple introductions of some of these invasives. And with more research and a paper written by Adrian Pender, but not this Adrian Pender, someone in the UK, but there was a very funny confusion for a little while there, is that Buddhist monks and the temples had their religious practices like to release fish into the environment, and it looks like maybe they're releasing some invasives, so we need to discuss about providing some data so they can release some better fish. So if we go to the next slide, so what we thought was one species of cichlid, goldfish, and common carp is probably actually more than that, which is not great. So if you could just go back, sorry. So the biggest group is the pearl cichlid at the top, and then it turns out that there is also two other cichlids, and then the goldfish at the top, carp, another carp, and the Russian carp. So if we go to the next one. So it could be that the other ones are in quite low numbers, but also some of them look very similar to each other. So a lot of people, unless like you're first spots and you're really great at IDing fish, we could be just thinking it's like, oh, this is a goldfish, because like the Russian carp and the goldfish, I can imagine people getting confused. So yeah, but as you can see, a lot of these are all aquarium species, and so yeah, people are releasing an integrated. Anyway, next slide. So what we really need to do is improve our reference library. So 80% of our sequence data we could ID as fish, but that's it. We couldn't go any further. Our pandanus and pandanus vistocchio, we cannot tell the difference between yet, because only one of the two species has sequence data in the reference database, and one's native and one's invasive. So we need to tell those apart. So big, and then frogs. Really no frog sequences on the database. We would really love to add those in. So yeah, a big thing we need to do now is just add more more sequences to the body of the moon. Paige, one of the Paige notes here, is working on that with me. She's doing a lot of sequence data and also doing a lot of sequencing of the invasives to try and work out how many species there really are, what's hybridizing, which is really what's going on, and try and also see if we can tease out if there's been multiple introductions or one introduction, if the barriers are actually working. We also need to do further sampling of the canning and other rivers and wetlands to see how things change over time, how they change seasonally, and just really what's going on. I'd just like to thank everyone who was involved in that project, particularly Bruce and Ashley at DBCA, Chris and Deepa for teaching me all about fish, from Vertebrates University for providing a lot of tissue samples for us to build our reference database, and the technical staff at WA who really helped Emma when she had to filter a lot of water. And everyone else, like Thomas Water and everyone else who happy. So that's it.
We do have about 3 minutes for questions before we need to reconvene.
Yeah, I've got probably a number of questions, but two main ones. One, the reference libraries, are they shared with all the universities and internationally so that, if you pick up sequences, that information is shared and you don't have to develop your own reference library and so on?
So the majority of the sequence library we get from public available databases. We're trying to develop a canning or like a WA specific reference library, and because we're a government department, everything then will be publicly available. Some things, like I think a lot of the frog sequences have been done, but they're just not available to people. But yeah, for the most part, things are publicly available, and then anytime any paper is published, then those sequences have to become publicly available. So it is happening, but it's just a lot of species down there, and it's just getting them all, and then there's just a lot of genes as well, that people pick different genes or different sections of genes, and so it's just trying to get it standardized and everything. So yeah, but it is happening and it is becoming more and more publicly available, and there's also a lot of sharing between people as well. But yeah, we are hoping to at least make a WA specific reference and then make that publicly available. That's the goal of the Pages project.
So my other question was, I work with endangered species that, there's less than 150 in WA, and so they're going from wetland to wetland to bird. So if, say, it went to wetland A 2 years ago, but it hasn't been back since, is there like a best before date for the eDNA?
So you want something to be long term, the sediment is where you go. You want something more recent and you want water. There's different sort of
Best before dates?
so like water has like a short term, leaf litter is a bit longer, sediment is longer, things in cave will last longer than things out in like the forest. So there's different ways of getting different times. And then like top surface of sediment is not going to last as long as you dig down and things like that.
This might be more than the client to be able, what are the costs associated with like eDNA? So I guess I see some wonderful applications from the local government point of view, which try to argue, against development happening or all sorts of things that we can try and look in that space. To try and figure out there's longer turtles in particular that we can't do traditional control, traditional cages or whatever. So ballpark figure, what costs are we talking about in terms of like laboratory work versus consultative collecting this citizen science point of view, collecting that information?
Really good question. And there's definitely economy and scale as well, because whether you're taking a batch of 10 samples through the lab or 100 samples, there's really not a big time difference. There are consumables and all of that, but the amount of time to put in. So, and then it really depends on the depth of sequencing you want from those samples and what it is you're asking about. Do you want that biodiversity survey, or are you only interested in looking for that one really critically endangered species, and does that occur there? So there can be some differences. Part of our lab, we have a commercial service, which is eDNA Frontiers, and Shane sitting right beside you. So in terms of cost, it's always best to reach out to Shane as well. Trend sits on the R&D development, so then again it comes to, well, what's the question again? Do we have the genetic assays available, or is that something that we need to develop? This is where our honors and masters and PhD students come in, because they work on these projects and then get them that.
You don't have the species in the database, is that what you mean?
It depends on what the project is. So yeah, usually we've got to do some desktop work first and go, all right, we're not up for recreating the wheel. If reference sequences are already available, then yeah, no, there's, we don't need to spend time on that. So that's what we usually do. We get a background on who's done what. And then we usually propose, all right, well, we were to do this project and it needs development and R&D and some validation in the lab, and that's what we can do. Sometimes we can support it in the student project or postdoc. If it's already
Sorry to interrupt, we're going to have to reconvene in the main audience. Everyone has an opportunity to ask questions during the dinner. Okay, so take them up.